Hello beloved students, today we are starting with the second lecture of immunology. So first of all we will discuss T cell and B cell. In the previous lecture we have already discussed the basics of the immunology. So T cell, the site of origin is bone marrow. In bone marrow they originate but they mature in the thymus. You can remember T for T. So thymus is the central organ. Central organ means organ of maturation. Organ of maturation is thymus. So T cell originate from bone marrow. They mature in the thymus. While on the other hand, B cell, they originate in bone marrow and mature in the bone marrow. So that is a simple classification of T cell and B cell origination. Now let's discuss how T cell and B cell detect the foreign antigen. So there is a very simple process that insoluble antigen can identify it by T cell receptor very easily. But soluble antigen cannot be identified by T cell receptor. So by what process the soluble antigens are killed, we will discuss in this topic. So for example, we have two cells. This is antigen presenting cell and this is T cell. This cell will present the antigen to this T cell. So let's suppose this is antigen presenting cell, and we have discussed in the first lecture that antigen presenting cells include dendritic cells, etc. So on this antigen presenting cell, there are two molecules. One molecule is called major histocompatibility complex 1 and other is called MHC2. Two molecules are present. On the other hand, on T cell, there are two receptors present. You can say that either you can say the two types of cells are present. One is called CD4 T cell, one is called CD8 T cell. Now, if this T cell is CD4, if this cell is CD4, the antigen presenting cell will express MHC2. If this T cell is CD8, the antigen presenting cell will express major histocompatibility complex 1 mean MHC1. So if antigen presenting cell will decide either to express MHC2 or MHC1, it will look towards T cell, whether T cell is CD4 or CD8. So this is called signal number 1. This is the first signal, and this is also called initiation signal. Now let's move forward. When antigen presenting cell express its molecule either MHC1 or MHC2, so then T cell is activated? No. T cell will require a second signal. And what is the second signal? On antigen presenting cell, on the other hand, there are another two molecules present. One is called CD80, one is called CD86. CD80 is also called B7.1 and B7.2. Now, when this CD8 or CD86, they combine with another molecule of T cell called CD28, the T cell will activate, and this signal is called activation signal. So for the T cell activation, two signals are required. The first is done by MHC and CD4 or CD8, and the second is done by CD80, CD86 with CD28, and this is called co-stimulatory signal. And the signal one was called, we call it initiation signal. So this is a co-stimulatory molecules, you can say, and this is a co-stimulatory receptor. Now T cell is activated. When T cell is activated, it will form cytokines, interferon gamma, interleukin 4, interferon gamma and interleukin 4. It will activate a B cell, and a B cell will express a molecule called CD40 ligand. CD40 ligand. It will express a receptor called CD40 ligand. Now when it expresses this receptor, it will combine with another receptor of T cell called CD40 receptor, and in this way this B cell will form. B cell will be converted into plasma cell, and when plasma cell is formed, it will produce antibodies. Antibodies production by this way. This I have make myself clear. Now, so one is T cell dependent method, one is T cell independent method. For example, we have discussed T cell dependent method, and there is another method, this is T cell independent method. But 90% cases T cell dependent method means that T cell activates B cell, and it is a major process 90%, and it have a memory, and it is a stronger process. While on the other hand, where T cell is not required for B cell production, it is less than 10%, no memory, and it forms a weak antibodies. Got these points? Very good. Now let's discuss another mechanism which is called energy, energy permanent inactivation of both T cell and B cell due to lack of co-stimulatory interaction. When there is no co-stimulatory interaction means when CD80 or 86 having no interaction with this co-stimulatory receptor, it means the cell will not activate it. In this process where permanent inactivation of T cell, it is called T cell and B cell, it is called energy, and it is due to co-stimulatory interaction. T cell energy permanent activation of both T cell and B cell. I have discussed permanent inactivation of both T cell and B cell due to lack of co-stimulatory signal. When this signal will not interact with this receptor, there will be permanent inactivation of T cell and B cell. This interaction will not develop. So, or you can say B cell when not express is CD40 ligand with CD40 receptor, the cell will also remain in permanent inactivation. So it is a mechanism where its application comes, where its use come. It's a mechanism to prevent autoimmunity in peripheral blood, and this process is also called peripheral tolerance. So I made myself clear. One is MHC restriction. I have discussed that CD4 will accept the antigen from MHC2 and CD4 will accept the antigen from MHC1. Good guys. Let's discuss classification of T cell. So T cells are broadly divided into two types, CD4 T cell and CD8 T cell. CD8 T cell is called cytotoxic cell, while helper T cell, CD4 T cell is called helper T cell. Helper T cell, and these constitute 60%, while this 30%. T cell, helper T cell is further divided into memory, effector, and regulatory CD4 T regulatory. Then effector is divided into T helper 1, T helper 2, and T helper 17. First of all, discuss a regulatory T cell. Regulatory T cell or CD4 regulatory. In peripheral blood, there is a presence of self-antigen. Let's suppose in peripheral blood there is a presence of self-antigen. So CD4 T regulatory express CD25 or it express FOXP3. So when it expresses these two molecules, they will synthesize anti-inflammatory cytokines 4, 6, 10 and tumor growth factor beta, and it will inhibit the proliferation and differentiation of autoreactive cell. Simply when CD4 T regulatory cell express these two molecules CD25 and FOXP3, there will be no interaction. There will be no interaction between the self antigen and the T cell. When there is no interaction, automatically there will be prevention of autoreactive cell proliferation and differentiation. Got this, guys? When there is a mutation in this gene FOXP3, so this mutation leads to a syndrome called IPEX syndrome, and IPEX stands for immunological dysregulation, polyendocrinopathy, enteropathy and X-linked. While on the other hand, CD25 mutation leads to a disease called multiple sclerosis. Got, guys? Now let's discuss Now let's discuss CD8 T cell and natural killer cell. Got, guys? So what is the mechanism of CD8 T cell? So for example, its mechanism is almost same. So we will discuss in a simple in the same slide, in a same topic or page. CD8 T cell and natural killer cell, they are having granules. When these granules rupture, they release an enzyme called purpurine and granzyme. This system, purpurine and granzyme. This purpurine goes towards the cell and they perforate the cell, means formation of the holes. And then this granzyme enter. When this granzyme enter, granzymes are of two types, A type and B type. The A form free radical while B express force ligand induction. Force or force ligand induction, it leads to apoptosis while it leads to destruction. While on the other hand, natural killer cell, when the granules release, same process, rupture, purpurine granzyme system, there will be one granzyme same form free radical while the other form what? The other form the same, force or force ligand and induction apoptosis. So simple process is followed in both the natural killer cell and apoptosis, natural killer cell and CD8 T cell. CD8 T cell is what? Cytotoxic T cell. Now CD8 T cell and natural killer cell, they have the same function, killing of virus infected cell, tumor cell and graft cell. If both are mentioned in the MCQ, so you will prepare CD8 instead of natural killer cell. Now let's discuss some knowledge about natural killer cell. CD16 and CD56 molecule, they bind to the fragment of immunoglobulin G. When this molecule binds to this antibody, it means that this killing is antibody dependent cellular cytotoxicity. Antibody dependent, because this molecule will bind to this antibody and then it will kill the cell. So antibody dependent cellular cytotoxicity is also present other than natural killer cell in eosinophil and neutrophil. Keep in mind. Now for example, this was a normal cell, this was a normal cell. When virus infect this, virus infection, when it is infected, so there is a reduction in MHC1. When there is a reduction in MHC1, it means that now inhibition is lost, and when inhibition is lost, now this natural killer cell will do what? It will form natural killer cell, and this is a virus infected cell. So when this is a, for example, this is a normal cell, and when there is an MHC1 reduction, natural killer cell will come and will eat this cell. Simple process. Got it, guys? This was a normal cell. This normal cell, when it was infected by a virus, there is a reduction in MHC1, and when there is reduction in MHC1, natural killer cell will be activated and will eat this cell. Now let's discuss about the effector T cell. Effector T cell. Effector T cells are of three types: Th1, Th2, Th17. Th1, it secretes interferon gamma, interleukin 2 and 12. Interferon gamma activate T helper 1, and T helper 1 itself secrete these molecules, and these molecules activate macrophage. These molecules activate macrophage, and the function of the T helper 1 cell is granuloma formation, intracellular killing, and delayed type 4 hypersensitivity. While on the other hand, interleukin 4, it will activate T helper 2, and then T helper 2 will secrete interleukin 4, 5, 6, and 13. Now, interleukin 4, it activates mast cell, 5 eosinophil, while 6 and 13 plasma cell, and function is in type 1 hypersensitivity reaction, parasite killing, and immunoglobulin E antibody synthesis. While on the other hand, T helper 17 associated with autoimmunity, it is activated by interleukin 1, 6, and tumor growth factor beta. It secretes interleukin 17 and 22. So the functions are severe inflammation, got guys, neutrophil and monocyte predominance associated with autoimmune disorders like rheumatic arthritis and psoriatic arthritis. Now, for example, this CD4 or CD8 cell, CD4 is activated by the expression presented by MHC2, while CD8 is activated in response to MHC1. But there are some molecules that they do not need MHC1 and MHC2 molecules. Instead, alpha beta receptors are present, and these kind of cells are called what? Delta gamma T cell. Delta gamma T cell. And delta gamma T cell is having no association with MHC, and they can combine with antigen without using MHC. So no need of CD4 or CD8. We have covered the topic. Hope you guys understood this topic. If you note the PDF form, I will mention my WhatsApp contact number here in the description or first comment box. You can contact me. And I think so this pathology we are covering from Robin and Levinson, as well as this immunology topic from Javed Immunology, one of the best immunology book, Levinson Immunology. So if you will concentrate, I hope you will understand these topics. If you are concentrating, you are created for a great purpose. You are doctors, and doctors not entrusted in earning. Doctors entrusting to serve the humanity, to guide the humanity. Because if you will become a psychologist, psychiatrist, you will guide the humanity. So I hope I made this lecture clear. And if you have any type of questions, you can put in the comment box. I mean, when you will go through this lecture, after this lecture, you will crisp and clear all these topics. I promise. Good guys.